muchas empresas farmacéuticas de alto nivel. Algunas de estas empresas están representadas también en esta sala. Y después estudió también o continuó con sus investigaciones en diversas ciudades de Suiza, pero también en Australia, donde conocí a su colega, el profesor Rolf, y también en los Estados Unidos. Ahora ha terminado su carrera académica o de investigación, pero aún comparte su conocimiento, toda su experiencia con los estudiantes en el hospital universitario de Zúrich. Entonces, soy realmente muy feliz de pasar la palabra ahora al profesor Rolf Zinkernagel. Profesor, por favor.
I thank you, Ambassador of Switzerland, Carol, very much indeed for your very kind words. About half of what you said is true, and I will try to live up to it. Now, it's a pleasure for me to be here with you, and I thank the Embassy and the Fondo Cultural, as well as the Commercial Association for having supported my travel to come here. Now, it's a pleasure for me to speak here because I'm considered a cultural product, which actually is not quite so often heard of. You know, science is culture. I think it's very much the same as artistic, concert, music, paintings, and other cultural parts of our life. And I think it's very important that we as scientists can transfer and transmit exactly that image, that science and research are as important as culture and art. And it has some similarities, and I will try to develop that concept. Now, I've just come down from the Andes after 2 weeks of vacationing. I'm retired, so I can enjoy life sometimes. And I've been to the classics, you know, Cusco. I've done my four days of trekking above 4,000. I've gone to Machu Picchu and Titicaca, and I've seen parts, of course. Not all. I would like to come back, and I'm particularly impressed by the Peruvian and, in general terms, you know, Indian culture of the slopes of the Andes with these terraces, with these various crops, and I think this is very fascinating, and actually is very similar to parts of Switzerland, particularly the valley. The valley's part is very much the same, you know, steep slopes, you build up terraces, you basically look that you can grow enough stuff to survive. So it's a great pleasure, and of course I have to ask Peter to quickly stand up so that everybody knows who Peter Doherty is. As you easily can see, he is my twin brother. We only worked for 2.5 years together in Canberra, and I'll try to sort of, you know, show you how that developed. But these 2.5 years, of course, have made us twin brothers, because our both lives really is linked to you know, discovering something nobody knew before we discovered it. And this, you know, is an essential time point and has since been for our lives, not only our own lives, also our wives and families, and even, you know, only having passed 2.5 years, as Peter usually says, we still talk to each other and enjoy it when we meet at occasions like this. Now, let me get slowly to the essence of matters. Now, research means looking again for something that apparently has already been known. Otherwise you wouldn't call it research, right? So we search for something that already is there, which in fact it is. We just don't know about many things in nature. And that's the essence of research in biomedical areas. To find out how biology, how nature works. And why certain diseases come up, and why some people are resistant to certain diseases, whereas others are highly susceptible. That's, you know, good for those who are resistant, let's say, to malaria. But of course, for those who are more susceptible, are weaker against malaria, it's bad. But those who have this, let's say, slight defect against malaria will be probably stronger against other diseases. So the risks of being, let's say, resistant or susceptible is simply distributed through the population. And usually these risks can be, you know, graphically be illustrated by a normal so-called Gaussian type of distribution. Very few are highly resistant, most are, let's say, plus minus okay, and few are highly susceptible. And for each disease, this sort of location is distinct, and therefore never all humans die of one disease. Some die, but the others survive. And so you get this genetic diversity distributed, you know, through the population, which basically makes us, overall, we do fairly well. Now, there is a very important observation that has been made over, let's say, the past 50 or 60 years. Because surgeons started to transplant kidneys if somebody had problems with the kidneys failing their function. So you had to undergo dialysis, which is a big machine that basically washes your blood. Or if you were lucky, you had a twin brother, which is rare, of course, who could give you one of his two kidneys. Now, why do I say twin brother? Because the twin brother, a true twin brother, is genetically identical. So, a true twin brother's kidney transplanted is accepted, is not rejected, because it's basically almost the same. But if you transplant any other kidney, for example, from a car accident or a motorcycle accident, within an hour or 3 hours, you can take out the kidney with the permission, of course, of the families and all the necessary cautions, and transplant that kidney. But then, because it's not genetically identical, that kidney is usually rejected. And this is prevented by pharmaceutical means of suppressing the immune response against these foreign kidneys. Now, the reason why a foreign kidney is rejected is that on the cell surfaces, on all our cells, we have certain molecules, certain structures that are called transplantation antigens. And they are called transplantation antigens because of that rejection mechanism that a kidney transplanted that does not come from an identical twin is rejected. Now, thinking about it, it's clear that is an experiment of modern medicine. This has nothing to do with normal biology, because up till 1950 there was absolutely never a transplantation of a foreign kidney. So these transplantation antigens could not have been invented by anybody to prevent kidney transplantation. So a question that arose in the 50s, 60s, 70s was simply, what the hell are these transplantation antigens for? They certainly have not been invented, as I said, to make life of transplantation surgeons complicated. And of course of the patients also. So that was about, let's say, the situation in the 1970s. There's a second observation, because now one started to determine these cell surface structures called transplantation antigens, and looked amongst, let's say, many potential donors of such kidneys, whether by chance one could find these antigens, which are, many antigens that are identical to the future recipient of such a kidney. And in fact, one in about 100,000 to one in about 1 million, you could find such a almost identical donor. And the more like the given kidney is with the receiving patient, the less likely is that immune responses kick that kidney out. So one gains by making it as close as possible. But while doing so, one found something completely unexpected. And that is that certain of these so-called transplantation antigens, of which we have, let's say, something like 10 on our cells, that some of these correlated with certain susceptibilities to disease. For example, if you have the surface antigen on cells called B27, it's not important we know what that is, but let's call it B27. Then you have about a 25 to 50-fold higher risk of getting certain rheumatoid types of diseases. Surprise! One didn't understand this correlation. Okay. Now, at that time, Peter was a crusty, young researcher, having reached Canberra in Australia back. He is actually born in Brisbane in Australia, so he did his PhD in Edinburgh, because at that time, still from Australia, Great Britain was sort of the Holy Grail. Like for me, Basel is the Holy Grail. So he came back to Australia and joined the Australian National University in Canberra, the John Curtin School of Medical Research. And I had applied for doing a postdoctoral fellowship to do research, because I had started to become a surgeon, and after about 1.5 or 2 years I thought, this is not going to end well, and I must look for a different type of medical activity. So I applied for a fellowship to go anywhere in the world. And I sent out about 50 applications. This was 1971. And of these 50 applications, 30 were never answered. And the other 20 were answered, but all negative. You're happy you don't come. And there was one exception. And this was that the head of department, the professor I worked for in Lausanne at the time, on questions of immunity against hepatitis viruses causing severe liver disease, and against and studying the effect of mother's milk on infections in babies. Actually mice, with the same problem. And he knew the head of department of microbiology at the Australian National University. So that opened a small gate for me actually to go to Canberra and work on problems of infectious disease immunology. So that's a chance event. I mean, had these two professors not known each other, I would not have landed in Canberra. And I wouldn't be here today. Next point is my wife at that time, also a medico. We had 2 kids at the time. We were about 28 or so. Could have said, I mean, are you crazy to go to Canberra? I mean, why should I leave Basel or Zurich to disappear into the Australian bush? No way. But she was courageous and, said, okay, let's go to Australia. So we actually went. And there I started working with one person within the department called Prof on some particular bacterial infections that are, serious health problems, like salmonella infections. These cause diarrheal, consequences, but also other bacterium called Listeria monocytogenes. That's the problem the Swiss had with some Mordor cheeses in the Jura Mountains, because they also can cause disease. So I worked on that, and there I got familiar with certain cells which you can isolate very easily from the peritoneal, the stomach cavity, let's say, in the mouse. And this has, will play some role later. But by chance, because the laboratories in Canberra are very small, they just can house 2 researchers, postdocs or whatever, plus a technician. So very small. I mean, you could probably put into this room 3 or 4 such units with all the equipment and everything. And the only space that was open was in Peter Doherty's, where he had worked previously. So, we smelled at each other. It's okay, and then he slowly started to work on problems. He still is a veterinary doctor. So he was interested in brain diseases of a special kind, particularly those that involve the lining of the brain, which are called meninges, and the infection of these linings called meningitis, which is a very serious life-threatening disease. But of a particular type, because not bacterially caused meningitis, which is, sort of the meningitis bacteria are clinically much dominant, like 90% of these infections come from bacteria. But a few are caused by viruses. And that virus basically does not do any harm if the infection is in a host, practically in a mouse, that has no immune response. And you would think this sounds strange, doesn't it? Because usually you think you need an immune response to get rid of the damaging virus. But in this case, the virus doesn't do any harm by itself. So in fact, it's paradox. It's the immune response that does the harm. And I will try to tell you, to illustrate this. See, this is a mouse. You infect it into the head with that virus, and then you see it makes some meningitis, and within 7 to 8 days the mouse dies. And if you take a mouse that has no immune response and you do exactly the same experiment, nothing happens. The mouse. So it's not the virus alone that kills the mouse. Actually, the virus alone doesn't do anything. It's the immune response against the virus that kills the mouse. And to explain that, I show you this sort of tissue section, which shows you that the soft lining of the meninges actually are the only cells that are initially infected. That's shown by the bright orange staining. So that's where the virus is. And now, I have to explain to you a test I learned in Lausanne when I was studying the other things. And this test was a very important test at that time because the T cell, the cellular immune response, was considered very important to protect us against certain tumors. But they also were found by the Lausanne group and some other Australians. The name is Jacques Miller. It was also shown that these cellular immune mechanisms also were largely responsible for the rejection of foreign grafts like kidneys or skin or hearts. And the test is illustrated at the left side. You see, you take cells and fantastic. You take cells and put them into a plastic dish. And then you take immune cells from the mouse and simply ask, can these immune cells lyse or destroy these cells you have grown in this plastic dish? And of course, cells from that mouse exposed to immune cells from that mouse shouldn't get destroyed, because the immune system doesn't react against our normal cells. It only reacts against transplanted kidney cells or skin cells. But it also, and that's where we were involved in defining, if you now infect these cells in the plastic dish with a virus that you have used to infect that mouse, then the immune cells from that mouse actually kill these cells in this assay. And you can measure that by putting something into the cells, as shown here, black dots. And if the cells die, the black dots get released into the recipient, and you simply measure, is it released or not released? And of course, the interpretation was very simple. If immune cells, T cells, can destroy either transplanted foreign cells or virus-infected own cells, then this could be an explanation for A, kidney rejection, but B, also how a virus-producing factory, which a cell is, you need a cell that is infected, that reproduces virus. Without a cell, virus cannot be reproduced. And if you kill that cell that is infected and produces virus, you stop virus production. So in a way, rejection of a foreign tissue graft like kidney is like rejection of a known cell that has been infected with virus. In the second case, it makes biologically sense because you stop the infections, whereas the case of the foreign kidney cells has never been a biological, really a biological. Okay, so now, we, and Peter was, probably still is, very dexterous in getting the fluid from the cushion around the brain, which is called cerebrospinal fluid. And in this fluid, because of what I've told you, this is in this space here, after an immune or inflammatory reaction, you see this is the normal condition, and after infection and immune cells invading, you have lots of cells in that area. And these areas you can also find in this fluid up here, and if you are as dexterous as Peter Doherty, you can actually get a very tiny little bit of that fluid out. And now you can repeat the test I've just shown. So you now take these cells and put them into this test. And what you find, in fact, if this mouse has T cells or an immune reactivity, these cells will destroy the virus-infected cells in this assay. Now, as I've told you, we all knew about this disease association that correlated with these transplantation antigens, which are called HLA in humans. It's human lymphocyte antigens. This is equivalent to these transplantation antigens. Unfortunately, in science, but I guess that's everywhere, you make life miserable and complicated by using cryptic names and variations of these things. This association was not understood at the time. So he said, if it is true that these cytotoxic or cell-destructive immune cells are responsible for this choriomeningitis and this early death after 7 to 8 days, then maybe we could vary these transplantation antigens, these so-called major histocompatibility gene complex antigens, forget it immediately, which are called in mice H2. We could vary those and find out, are certain types surviving, not killed at all by this immune process, while others are get a disease and get killed very early. So it was basically a reasonable idea. But when we did that experiment, and this is the first experiment we did, we have several different mouse strains. We found that after 7 days of this infection, that in this, release assay that I've shown you, that some will release uninfected, but not on normal uninfected cells, whereas other mice lymphocytes will not release differentially between infected and uninfected. You would say this is not 0, it's 70. But this is just the spontaneous sort of release of these black dots I've shown you, which you cannot stop. So you could correct for that, but that's not important. The difference here is that these lymphocytes release or kill these target cells, these don't. But if you take a breeding between the mice that make lymphocytes that can kill these infected targets with a mouse father that cannot kill, then you find again a positive result. And going through all this, it was very clear that whenever we had a K in this transplantation antigen type, we saw a positive result in the test. So that was observation 1. Observation 2 is we left a few of each of these mice going after the infection. Remember, if you put the infection into the cranium, into the head, then the mice die if they have these cytotoxic or killer T cell immune response. So we tested this in our assay, in vitro, in these plastic wells, but we also looked at the clinical development of the disease. And all the mice, irrespective of whether they were positive or negative in the test, they all got killed. They all died. On day 8 or 9, they were all dead from the disease. So we had a discrepancy. On one hand side, we had the clinical observation. All the mice suffered from the disease and died. And we had a test where only some were positive and others negative. What do you do? Well, you can do two things. You can say, you know, the test is the right thing. But, you know, me being a medico and Peter being a vet, it's always the patient is right. Because if the patient dies, something is wrong. And it apparently isn't the disease that is wrong, because it killed all mice. So it must be the test. But then you have to make a decision. So we made the decision, it's the test that is somehow faulty, or not correct, or not correlated. And then it was very clear, because by chance, the cell lines that we used in this plastic, you know, dish that we infected or left uninfected were derived originally from a mouse called C3H, forget it, but it has the characteristic of K on the surface. And you remember that mouse is a K, and that has a K. And those were the two positive test cases. So there is a correlation between what the tissue cultured cell has on its surface and what the immune responding mouse has on its surface. So the conclusion was, and we then did follow-up experiments, was the immune cell that kills the target cell in that in vitro assay somehow must recognize two things. A, that it is virus, but B, it also must recognize that it has the K on the surface. Otherwise, it wouldn't touch it. Because immune cells from the C. B mouse or the C. black B mouse would not touch these K infected targets, although we used the same virus to infect all these mice. So the virus alone is not good enough for this cell-destructive immune reactivity. So then, of course, we thought if that is so, then we do have an explanation for the function of transplantation antigens, because it now looks as if exactly the same structures, the transplantation antigens, are responsible not only for foreign tissue rejection, but also for the recognition and rejection of our own cells, mechanism unknown. So we did, of course, this symmetrical experiment. We now use target cells, let's say, of D type, infected those with the virus and tested immune cells from a D mouse, and we got lysis. So what is negative on K infected cells is in fact positive on D infected cells. So there's a clear necessity for these immune cells to not only recognize the cell surface characteristic KD or whatever it may be, they also must recognize virus. They cannot recognize virus alone. They only recognize a combination of a self structure, DKB, and this foreign introduced structure that seems to combine somehow with these transmutation antigens. Again, mechanism unknown. We then made predictions or modelings, thought, you know, how could one explain that? And by good luck and good fortune, we made some predictions which then were demonstrated by other people, not by us. And this may be a bit too complicated, but this is, you just imagine that this structure here, this is a molecule that sits in a cell membrane here, and this molecule has like a presentation area here. And into that presentation area, a small bit of the virus is sort of, you know, grabbed and presented on the cell surface. So that structure picks up a little bit of digested viral parts and brings it onto the cell surface. And because that sausage here in the middle, when we look from the top onto this structure, this little sausage has not been in the host before. It has been brought in, so to say, via the virus infection. This combination is recognized by the immune cells. This is work by a group in Oxford. They defined the quality of the sausage, and some biochemists and molecular biologists who were particularly interested in the structure of these transplantation antigens, names such as Wiley or Björkman are associated with this structure. So this explanation came later, about 10 years later, after we had found the original observation that immune cells do not just recognize something of the virus, but a part of our own, let's say, cell surface where some bits of viruses somehow associated. Now that's basically it. You know, this was done in the first 8 months after I came to camera, and it only was possible because we worked together, and each one of us would not have found what we found together. And this was then a free ticket to Stockholm about 24 years later. And I tell you, the Swedes can really make a nice celebration, and we enjoyed that with our families extremely. So, what are my general conclusions? You know, to find a disease that intrigues you and challenges you to find out why does this disease come about? How can we understand the process? I think that's number one. Because unless you are extremely happy with the question, and you really want to pursue it, you get frustrated. Because what sounds relatively simple, you know, everything goes wrong that can go wrong. I mean, you know this principle. So there's a lot of frustration, but sometimes you hit it lucky. And this was certainly one time. So you have to like the question to survive the frustration. Secondly, you have to work hard. You know the American saying, the harder I work, the luckier I get. It's true in science as well. Then a little bit of intelligence helps, because you have to be awake when you see the discrepancy. Because there are many other ways to deal with this discrepancy. You simply say, well, the test didn't work, you know, or I didn't count correctly, or the sun was too bright, or something like this. Then you need good luck. I mean, I've tried to sort of follow some of these chancy events. I mean, just think, Catherine, my wife, would have said, forget it, I don't want to go to Australia. This would not have happened, at least not by us. The good thing in science is, if Peter and I wouldn't have done that type of experiment and wouldn't have found this sort of discrepancy, somebody else at some time later would have found it. Because it's an important principle and somebody would have found it. So we just were really very lucky. I think it's important to have some concrete, tangible biology, such as a disease or a death, because it motivates, I think, anybody, particularly medicos and vets, to go after the reasons for the disease. I think to have discrepancies is always a challenge. You either can confront them, I think in business it's exactly the same, or you can avoid them, and this usually is not a very good strategy, because important problems and the important questions don't go away. If it's a lousy or an unimportant question or problem, they usually go away, but that's why they are unimportant. And I think the last thing is that we still have to be aware that we know more than we used to know 20 or 40 years ago. That's so-called progress. But we still know very, very little. And if you find something that really changes the outlook or the concept or the understanding of something, then, you know, that's a very good and very nice moment. That's when you enjoy it. It's like a good concert, you know. It's fantastic to find out how things work. And that we had the privilege to experience that, that's of course great. And then all the rest, you know, is frosting on the cakes, as the Americans say. It's nice to have, but the essence is to have made such an experiment, because nobody can take that away from you. But that's like in good business, or in good whatever. If you do something unique, courageous, you know, together with luck and intelligence and hard work, you still enjoy having found something that nobody else has seen before. Thanks very much.